In this short lecture, I'm going to discuss a number of primary immunodeficiencies. We have discussed a lot of these before in the class, and you'll notice that a lot of these immunodeficiencies really are going to review many of the basic immunology mechanisms that we have discussed thus far. So as you study these immunodeficiency diseases, you may wish to take an opportunity to strengthen your understanding of some of the basic mechanisms underlying the immune functions related to these diseases. I'll start first with deficiencies of the adaptive immune system. I'll begin with the discussion of severe combined immunodeficiency, then B cell or antibody deficiencies, and last T cell deficiencies. Now, severe combined immunodeficiencies can be most simplistically described, perhaps a little simplistically, as simultaneous loss of B and T cell function. And that can mean a complete absence of B cells or B cells that are present but can't make antibodies. It can mean a complete absence of T cells or T cells that are present but that can't make cytokines or kill their targets. One of the things to note about SCID is that it almost always results in defects in early development of lymphocytes in the bone marrow. And there are a number of features that are kind of tells as far as when you might be seeing a SCID case. One is that there's recurrent severe infections, and that starts right at birth. These infections have unexpected severity and frequency. There can be opportunistic infections involved, and infants with SCID often have a failure to thrive, and this is a fatal condition if untreated. Stem cell transplants are an option to treat individuals with SCID, and there are experimental approaches these days using lentiviral and retroviral vectors to promote corrective gene therapy. Now, the most common form of SCID is X-linked SCID. And to describe X-linked SCID, I'm first going to talk about the interleukin-2 receptor. Activated T cells express the high-affinity interleukin-2 receptor. This is composed of interleukin-2 receptor alpha, beta, and gamma. It's a trimer. And when interleukin-2 binds this receptor, it induces clonal expansion. Now, developing lymphocytes in the bone marrow don't express the high-affinity IL-2 receptor, but they do express one component of that receptor, and that is the IL-2 receptor gamma. And the IL-2 receptor gamma is commonly referred to as the common gamma chain, and that's because it's a shared component of many different receptors for different cytokines, including the IL-2 receptor, the IL-4 receptor, IL-7, IL-15, IL-21 receptors, and so forth. In this case, it's important in common lymphoid progenitors because it pairs with IL-7 receptor alpha to form the IL-7 receptor and induce signaling when it binds to IL-7 that promotes survival and proliferation and differentiation of these progenitor cells. Now, if you lose IL-2 receptor gamma, which is an X-linked gene, you impair IL-7 signaling during this process, and this absence of IL-7 signals leads to impaired development of these lymphocyte populations. And X-linked SCID is noted by the absence of T cells and NK cells, natural killer cells, which is another lymphocyte population but is part of the innate immune response. And B cells actually may be present in this disease, but they can't make any immunoglobulins. So again, X-linked SCID is an absence of the gamma portion of the IL-2 receptor, but has nothing to do with IL-2 signaling. Rather, it's a defect in IL-7 signaling. Now, there are autosomal recessive versions of SCID. One common autosomal recessive version of SCID is that induced by loss of adenosine deaminase, also loss of purine nucleoside phosphorylase. These are molecules that are kind of scavenger molecules that clean up toxic metabolites and prevent them from killing lymphoid progenitors. But in their absence, these toxic metabolites can lead to progressive loss of B cells and T cells and NK cells. And another autosomal recessive version of SCID is loss of the RAG recombinase gene. And if you'll recall, RAG is a recombinase that induces recombination of the V D, and J segments during lymphocyte development. In the absence of RAG, you get no antigen receptor rearrangement. These B and T cells undergo apoptosis. You do have NK cells present, however, because of course they don't undergo antigen receptor rearrangement. Sometimes this is called Omenn syndrome, and I should mention that all of these forms of SCID, X-linked and autosomal recessive, are quite rare. Now, there are a number of antibody deficiencies as well that one should know about. There are some key features of antibody deficiencies that I want to highlight for you. One is that these deficiencies often don't begin their onset until after 4 to 6 months of age, and that's because that's about the time frame in which maternal antibodies wane. Like SCID, you can get many recurrent bacterial infections with antibody deficiencies, and these infections have unexpected frequency and severity or unexpected complications. Common features of these bacterial infections are sinusitis, otitis, bronchitis, and pneumonia. And you can be susceptible even to unexpected vaccine-preventable infections. So you might have been vaccinated for this, and then you still get this disease for which you were vaccinated. One of the primary antibody deficiencies is X-linked A-gamma globulinemia. Now, this is related, something we discussed when we talked about lymphocyte development. But I'm going to go back and just review some of the biology here. So when heavy chains during lymphocyte development successfully rearrange, they get expressed at the cell surface, and they often pair with this conserved molecule called the pre-B cell receptor that stabilizes the expression of this heavy chain at the cell surface. And when this heavy chain gets expressed at the cell surface, this induces an intracellular signal mediated in part by this kinase, Bruton's tyrosine kinase, that tells this cell to survive and prevents it from undergoing apoptosis. Now, if the heavy chain fails to get expressed at the cell surface because you didn't have a productive rearrangement, then you don't get this survival signal induced by Bruton's tyrosine kinase, and this cell undergoes apoptosis. So this is a failure to undergo positive selection. Now, what happens in X-linked A-gamma globulinemia is you lose Bruton's tyrosine kinase, which is the specific mutation in this disease, and therefore you lose this survival signal, and this cell instead undergoes apoptosis whether or not it has generated a functionally competent heavy chain at the cell surface. And so the result is you get no mature B cells and no immunoglobulins. You get these recurrent infections. You can even get some chronic viral infections in the case of this disease. Okay. There are also antibody deficiencies associated with activated B cells. And one of the primary of these is hyper-IgM syndrome. Hyper-IgM syndrome results from a lack of CD40 ligand expression by follicular helper T cells. And so here's a T cell that normally would be inducing help to this B cell through expression of CD40 ligand. But in hyper-IgM syndrome, it is unable to provide this co-stimulatory signal. And this results in poor activity of activation-induced deaminase, which, as we know, is responsible for affinity maturation and isotype switching. You fail to form germinal centers in patients with hyper-IgM syndrome. And these patients generally will have high titers of IgM and low titers of everything else. One of the key features is that they have a very poor vaccine response. And also, for reasons that aren't entirely clear, patients with hyper-IgM syndrome have neutropenia. They do have a similar susceptibility to severe bacterial infection as other Ig deficiencies, but they also have this additional susceptibility to some opportunistic infections. And I'll mention that, I'll explore that in just a moment. And lastly, I do want to mention there are autosomal recessive versions of this disease, such as deficiencies in CD40 and activation-induced deaminase. And the reason why there's an increased susceptibility to even opportunistic infections in this antibody deficiency is that T cells also provide help via CD40 ligand to macrophages. And so this co-stimulatory pathway is important for macrophage activation, and you can have heightened susceptibility when this pathway is absent. And so one of the key infections associated with X-linked hyper-IgM syndrome is pneumocystis jirovecii pneumonia. Okay. There are a number of other antibody deficiencies. I'm only going to mention two briefly because you will have heard some about these in Dr. Kobernick's lecture. One of these is combined variable immunodeficiency. This is a deficiency that can sometimes onset later in life, the second or third decade. 10% of CVID cases result from defects in a receptor called TACI or TACI. This is a receptor for the cytokine B cell activation factor. But the fact is that the vast majority of cases, there's no known genetic basis. And this is a variable disease as indicated in the name. Sometimes this can result in an absence of all antibody isotypes. Sometimes this can result in an absence of one or two antibody isotypes. And if you have this disease, you're susceptible to recurrent and chronic infections and require immunoglobulin infusions to ward off these infections. IgA deficiency is probably the most common immunodeficiency. 80 to 90% of individuals with IgA deficiency are asymptomatic. But if you are symptomatic, you can have some recurrent infections and including some chronic diarrhea problems, for example, with Giardia. And IgA deficiency can progress to CVID and is linked to an increased risk of autoimmune disease, although the mechanisms behind that are entirely unclear. Now, moving on to T-cell deficiencies, one key T-cell deficiency that I'll mention in this course is DeGeorge syndrome. And I've mentioned it before during our lecture on lymphocyte development. This is a specific chromosomal deletion 22q11.2. And there are, this can lead to partial or complete failure of thymic development. Partial DeGeorge syndrome is much more common. Only about 1% of DeGeorge cases are have complete lack of thymic development. And when that happens, you have a complete absence of T cells. There's some other key features you'll want to learn a little bit more about as you learn more about DeGeorge syndrome, although they're not specifically immunology features. Patients with this often have hypoparathyroidism and hypocalcemia and congenital heart defects. It's common to have these kind of heterogeneous facial deformities, as I'm showing here, such as a rounded jaw, cleft palate, and other facial deformities. And these patients, especially if you have complete DeGeorge, you can be susceptible to severe viral, fungal, and mycobacterial infections. And there are some experimental approaches, such as thymic transplant, that can be attempted for patients with complete DeGeorge. There are a few other congenital deficiencies that impact the adaptive response. We're not going to talk about them much in this class. I just want to highlight them for you so that as you start preparing for boards later on next year, you'll be aware of these diseases and can circle back around to learn more about them. Wiskott-Aldrich syndrome is a defect in cytoskeletal structure. Ataxia telangiectasia is a defect in double-stranded DNA break repair. Chronic candidiasis can result from defects in IL-17 secretion or Th17 development. There's hyper IgE or Job syndrome that can result from defects in a transcription factor called STAT3. Loss of this transcription factor results in a loss of Th1 and Th17 differentiation, and this in turn kind of leaves a vacuum where hyperactive Th2 responses can take over. And you can get other defects that can result in lack of MHC class 1 or MHC class 2 expression. So that's it for deficiencies of the adaptive immune system. I want to briefly review a few immunodeficiencies of the innate immune system. We've talked about most of these immunodeficiencies before, so hopefully this will just be review for you. First, there are complement deficiencies, and these can be deficiencies in the classical pathway, which can lead to susceptibility to infection with Neisseria meningitidis, an encapsulated pathogen, other encapsulated bacteria as well, such as Streptococcus pneumoniae and Haemophilus influenza. But really, when you're thinking about complement deficiency, you're really thinking primarily about susceptibility to Neisseria meningitidis infection. Defects in the lectin pathway, I'm sorry, that says alternative pathway, but it should read lectin pathways such as MAS proteases and mannose binding lectin, and in the alternative pathway such as factor D and factor B, as well as C3, although these mutations are quite rare. There can be defects in the membrane attack complex that lead to defective formation of the MAC, and there can be defective mutations leading to defects in inhibitors of complement, such as factor H and factor I. And the interesting thing about these mutations is they also lead to susceptibility to Neisseria meningitidis. And the reason for this is by failing to regulate complement, they lead to overactive C3 convertase activity, and this in turn leads to consumption of C3 and depletion of C3 from the serum, so that you no longer have available C3 to respond to infectious insults. And so defects in many of these inhibitory pathways have the same disease susceptibilities as defects in the complement activating pathways. Another complement-related disease is paroxysmal nocturnal hemoglobinuria. This results from mutations in the PIG-A gene, which forms GPI anchors for two inhibitory molecules, CD9 and decay accelerating factor. And so in the absence of PIG-A, these molecules are no longer tethered to the cell surface, and so this leads to overactivation of the membrane attack complex. And again, you can have heightened susceptibility to Neisseria meningitidis when you get this disease. And I should mention this is a weird disease because it's an acquired mutation. It's on the X chromosome. It's not inherited. There aren't very many diseases like that, but this is one of them. Hereditary angioedema is due to a loss of C1 inhibitor. Again, we've already discussed this disease. I just wanted to reiterate the fact that this actually has nothing to do with complement, because C1 inhibitor inhibits a variety of different pathways in the coagulation, fibrinolysis, and contact system, as well as in complement. And in this case, overproduction of bradykinin can lead to this angioedema event where you get this rapid swelling of hands and feet and face. Here's a couple of pictures of people undergoing kind of this acute edema, and this can become more severe in late childhood and adolescence. Okay. There are other defects in the innate immune system, such as chronic granulomatous disease. Chronic granulomatous disease is a result of defects of the NADPH oxidase complex, and the X-linked version of this disease is specifically a defect here in GP91. This GP91 subunit is required for the redox activity of this NADPH oxidase, and so in the absence of it, you don't get redox activity. You have reduced oxidative burst, reduced reactive oxygen species, and of course impaired phagolysosomal function. And this impaired function results in a failure to control infection and leads to the formation of granulomas. And the granulomas are often look something like this, where they have macrophages in the middle and rings of Th1 cells on the outside, and kind of this active infection simmering within the middle here. And so this is a disease in which you are more susceptible to a number of severe bacterial and fungal infections. And the last one I'll talk about is leukocyte adhesion deficiency. This is the loss of LFA1, the integrin LFA1 on the surface. And as you'll recall, LFA1 activation, or the induction of this high affinity LFA1, is required for really all leukocytes to firmly adhere to endothelial cell walls so that they can then migrate into tissues. In the absence of LFA1, this firm adhesion never happens. And so you end up with neutrophilia in the blood, but very few leukocytes traffic into tissues and become susceptible to a number of severe bacterial infections. Okay, that's it for our review of immunodeficiency diseases. Again, use this material to review many of the basic mechanisms of how the immune system functions.